Hey everyone, this is Elad from Astrolabe Diagnostics, and today I would like to discuss the quality control aspects of Astrolabe's report export. So part of the Astrolabe platform is that you can download all of the analysis that Astrolabe has done in various formats. For example, you can download FCS files, you can download R files with single cell data, and finally you can download one big export called reports, and that includes spreadsheets, PDFs, and figures that cover every part of the analysis. One of these is different quality control measures, and I'm going to demonstrate them now. So this is how the report export looks like. You get it as a zip file, and just to give you a sense of scope, it has a lot of information. This is 3 gigabytes with almost 3,000 files. So there is a lot here to parse, and I'm going to focus again on quality control. So first of all, this is site of data, and site of data has beads. So by clicking on the beads directory, you can check out the bead gate that Astrolabe has created. To clarify, this is done using clustering, and this is using all of the bead channels you have in your experiment. So you know that the events that Astrolabe identifies are indeed beads. The next part is called non-cells, and this shows the debris and doublet process that Astrolabe has done. So what we see here are 3 different plots. We're looking at event length versus DNA, and we can see the cell population, the debris down here, and the doublets. Notice that Astrolabe's cleaning algorithm actually incorporates marker expression data as well. So even though we're seeing just event length versus DNA, this is following convention. Under the hood, the platform is using the different antibodies too. Another view you can see is what we call cell labeling debris. And these are cells that essentially slipped through the cracks of the labeling algorithm. And we see 2 of them here, canonical marker negative and unassigned. These respond to cells that did not express any of the major lineage markers and cells that express too many of them. And essentially, they slip through the cracks of the cleaning algorithm. You see that from event length and DNA perspective, they are in a pretty good spot. However, they are pretty suspect, and you might want to remove them from your analysis. This is your choice. Finally, one more thing you can examine here is the labeling hierarchy that Astrolabe has created. In other words, how did Astrolabe identify the different cell subsets in your experiment? This PDF is structured to be included as a supplement in your paper. It covers the algorithm, it covers different steps, and you can use it to navigate through the different cell subsets. So for example, if we click on B cells, Astrolabe will inform us that B cells are positive for CD19, negative for these markers, and you can examine the biaxial plots for this population. We can dig deeper, so let's say we want to look at CD27 positive B cells. This is the CD27 positive versus negative. And finally, if we want to look at plasmablasts, we can click on them here, and Astrolabe will inform us that it found 10 cells that fall under the plasmablast definition in this file. So I hope this was a helpful review, and also an important reminder that every aspect of your experiment is logged, documented, and reported by Astrolabe. There's no proprietary algorithms, there's no black boxes, everything is fully transparent. If you need any help analyzing your data, please reach out. I'm Elad at astrolabediagnostics.com, and I'm happy to discuss your analysis challenges and how Astrolabe could help. Furthermore, please remember to subscribe to us on LinkedIn and Astrolabe, on LinkedIn and on the Astrolabe YouTube channel. Thank you, and have a wonderful day.